Good day, students. Welcome again to our science class. This is again Ma'am Jen, and I will be discussing to you today the topic on your Module 1, Week 4 to 5. Okay, so let's get started. Okay, so before we will start our topic today, let's have first our unlocking of difficulties. So we will define all the terms that we will encounter as we go along with our topic today. Okay, first we have genetics. It is a branch of biology dealing with the study of heredity and variations. Next, we have heredity. It refers to the transmission of traits from parents to offspring. And then variation. It refers to the differences between individuals within species. Then we have the gene. It is the basic physical and functional unit of heredity. And then we have allele. It is a variant form of a gene. And then we have genotype, the genetic makeup of an organism. An example of a genotype is a 4 o'clock flower. The genotype of the red one is FRFR. And for white is FWFW. And now, let's proceed to the next term, which is the phenotype. When we say phenotype, it is the physical appearance of an organism. Okay, let's have again our example, the 4 o'clock flower. The phenotype of the first flower is red, and the phenotype of the second flower is white. And now for our next term, we have homozygous. It means that the organism has two copies of the same allele. Example, we have here for the height phenotype. We have tall and short. The genotype of the tall trait is TT, or 2 capital letter T. This is what we call as homozygous because the two alleles are identical, or the two letters are written in capital form. Then for short trait, its genotype is small letter t and another small letter t. Or in short, this is homozygous short and the other is homozygous tall. And for our next term, we have heterozygous. It means that the organism has two copies of different allele. Example, we have here again for the tall trait. Another genotype for the tall trait is capital letter T and small letter t. So this boy is a heterozygous tall, meaning he is not pure tall. Next term, we have dominant trait. It is a trait that masks the expression of the other. Example, this heterozygous tall boy. He is tall because being tall, which is being represented by capital letter T, is dominant over short, which is being represented by small letter t. Our next term is recessive trait. It is a trait that is being masked by the dominant trait. Example, we have this short boy. He is short because being short is recessive. Next, we have Punnett square. It is a graphical representation of the possible genotypes of an offspring from a particular cross. Then we have Gregor Johann Mendel. Gregor Johann Mendel was an Austrian monk who discovered the basic principles of heredity through experiments in his garden using the pea plants. He proposed the 3 laws of inheritance, namely the law of dominance, law of segregation, and the law of independent assortment. Through the years of many researches and theories of the different scientists all over the world, His research and theories are considered fundamental to any understanding of the field, and he is thus considered the father of modern genetics. Next, we have non-Mendelian pattern of inheritance. This is a type of inheritance wherein the pattern of phenotypes does not coincide with those that was presented in the Mendelian law of inheritance. And this is our topic for today. There are three types of non-Mendelian pattern of inheritance. So let's discuss first the incomplete dominance. It is when a dominant allele does not completely mask the effects of a recessive allele, and the organism's resulting physical appearance shows a blending of both alleles. Okay, so we have here example of incomplete dominance. A red snapdragon flower is crossed with a white snapdragon flower. The resulting offspring is a pink snapdragon flower. Why is this so? Now let me show you how this happened using our Punnett square. So we have here the genotypes of the two parents. For the red flower, its genotypes are FR and FR. And for the white flower, its genotypes are FW and FW. So when we say genotype, it is a pair of genes or alleles or a combination of two genes since they are from both parents, the mother and father. So, after identifying the genotypes, the next step is getting the gametes or sex cells. So, in getting the gametes, we'll only have to segregate the pair of alleles of each parent. Okay, for our red flower, we have FR and FR placed here at the top of the Punnett square. And for the white flower, we have FW and FW placed here at the left side of the Punnett square. Okay, next, let's have our first cross to have our first offspring, okay, or child. Then, okay, this one and this one. Okay, then this one for our second child or second offspring, this one and this one. Okay, so after crossing, the resulting offspring are, okay, we have FR, FW for the first offspring and another FR, FW for the second offspring. Okay, and for the third and fourth offspring, we have again the same genotypes, which are the FR, FW. So as we noticed, the four offspring have the same genotypes. So the resulting genotype of the four offspring of the red and white parents is 100% FR, FW. And the resulting phenotype of the four offspring is 100% pink. No one is red, no one is white. All are pink. Okay, so let's have another example. What are the possible genotypes and phenotypes of a cross between red and pink flowers? Okay, so let's get the genotypes of the two parents. For white flower, its genotypes are FW, FW. Then for the pink one, its genotypes are FR, FW. Okay, next is getting the gametes. So in getting the gametes, we will only have to segregate the pair of alleles. Okay, for the white flower, the gametes are FW, FW. Then for the pink one, the gametes are FR, FW. Okay, so let's start now our cross. For our first offspring, its genotypes are FR, FW. Then for our second offspring, its genotypes are FR, FW. Then for our third one, its genotypes are FW, FW. Then for our fourth one, its genotypes are FW and FW. Okay, in getting the genotypes or the genotypic ratio, we have 50% FR, FW, or 2 out of 4 offspring. Then 50% FW, FW. Again, 2 out of 4. Then for the phenotypes or the phenotypic ratio, we have 50% white and 50% pink. Now let's move on to the next type of non-Mendelian pattern of inheritance. Codominance. Both alleles are expressed equally in the phenotype of the heterozygote. Example, black chicken is crossed with a white chicken. The resulting offspring is checkered chicken. Okay, let me show you again how this happened using our Punnett square. Okay, so identifying first the genotypes of the two parents. So we have for the black chicken, its genotypes are CB and CB. Then for the white chicken, its genotypes are CW and CW. Okay, next is getting the gametes. So for the black chicken, the gametes are CB and CB. Then for the white chicken, we have CW and CW. Then for the offspring, we have, okay, so for the genotypes, 100% CB, CW. Then for the phenotypes, 100% checkered chicken. Okay, that ends our discussion. Thank you for watching.